Good morning, students. Today we are continuing with this chapter, Sexual Reproduction in Flowering Plants. In that chapter, today we are discussing the entering of pollen tube into a synergid. So, how the pollen tube will enter into the synergids and what are the events occurs in the synergids to releasing after the releasing of male gametes into them. So that we will discuss here. From that, in some plants, the pollen grains shed in two-celled condition, that is vegetative cell and generative cell. In such plants, the generative cell divides to form two male gametes during the pollen tube growth in the stigma. In other plants, pollen grains are shed in three-celled conditions where the cell containing, the pollen grain contains two male gametes at the beginning. After the entering of pollen tube reaches to the ovary, then to the ovule and reaches to the synergids where they release the male gamete. So, in the cytoplasm of synergids, two male gametes are released through the micropylar end of synergid, where they entering this pollen tube and they release the male gametes. Here at the micropylar end of synergids, where a finger-like projections observed when we studied at the structure of embryo sac of female gametophyte. At that time, we can know that another structure will be observed onto the synergids at the micropylar end, that is called as the filiform apparatus. The filiform apparatus present in synergids at the micropylar part, where this filiform apparatus will guides the pollen tube entering into the synergid. So, the function of the filiform apparatus is to guide the pollen tube for entering into the synergids. Remember this function. Now, here you can observe with the help of diagram how the entering of pollen tube into the synergids. So, at the micropylar end, the pollen tube will be enter inside the synergids through the filiform apparatus. You know that this is the central cell, these are the polar nucleus, these are the synergid, this is the egg cell and the nucleus. Here at the filiform apparatus in the synergids, they are guiding this pollen tube entering into the synergids. Here within this pollen tube there is a two male gametes are observed and one of the vegetative nucleus. Vegetative nucleus means the cell of the pollen tube where the vegetative nucleus is present. After that, in second diagram you can observe how the discharge of male gamete into the synergids. So after this is the filiform apparatus will guide the pollen tube entering into the synergids. In this cytoplasm of the synergids, the male gametes are released. So male gametes are released in synergid, one of the synergid, another male gamete will enter into the egg cell. So here a plant breeder which can manipulate, a plant breeder can manipulate the pollen pistil interactions. The pollen pistil interactions even in the incompatible pollination, means incompatible pollen grains also stick on the stigma. At that time also they can manipulate to breeding that pollen tube entering into the stigma, and the male gametes fuses the nucleus of the polar nuclei as well as the egg nucleus. So this is about how the pollen pistil interactions in that the entering of pollen tube into a synergids. After this we can discuss the artificial hybridization. Artificial hybridization is another technique used by the plant breeders. Artificial hybridization. Artificial hybridization, it is the crop improvement technique used by the plant breeders where the desired pollen grains are pollinated by artificially. The desired pollen grains are used for pollination. So, artificial hybridization, it is another technique in which the pollen grains desired pollen grains are used for pollination. So, it is the technique used it is a technique used for crop improvement. Program. It is the technique used for crop improvement program where pollen grains that to be desired one. So desired pollen grains. So desired pollen grains are used for pollination. So you can observe that artificial hybridization, it is a crop improvement program or the crop improvement technique which is used for the pollen grains, where we use the pollen grains which are desired one. So desired pollen grains are used for pollination. So the technique which is used for making the crop improvement, they are utilized the desired pollen grains for pollination. In this technique is achieved by following steps. There are following steps are there. Following steps. First one, emasculation. Emasculation, what is meant by emasculation? Here in this technique, in this step, the removal of anther, from the bisexual flower. It is a technique where the removal of anther from the bisexual flower in the female parent, that is female parent flower. So we can denote in this technique male flower parent and the female flower parent. So in female flower parent, where we remove the anthers in bisexual flowers, after that removed anther from the floral bud, that is bisexual floral bud of female parent, it is bagged by the help of the technique called as bagging. So first step, where the anther is removed, from bisexual flower of female parent. Bagging, after the emasculation, the emasculated flower is bagged. It is covered by a bag that is made up of by usually the butter paper. So bagging technique, where the emasculated flower, of female parent covered by bag which is made up by usually butter paper. So emasculated flower in second step is covered by bag that is made up by butter paper. Why this bagging means? To avoid the contamination of stigma of the female flower. To avoid the contamination of the stigma by unwanted pollen grains. Through the unwanted pollen grains, this emasculated flower is protected. So, the first step in that artificial hybridization, emasculation, where anther is removed from the bisexual flower of female parent. After the emasculation, the emasculated flower of a female parent is covered by a bag which is usually made up by butter paper. For protecting or for avoiding the contamination of unwanted contamination of stigma of a flower from the unwanted pollen grains. So, bagging technique, in that first you bag over the emasculated flower. Second, to avoid that emasculated flower to contaminate it from the unwanted pollen grains. This is the second step. After that, third step is the artificial pollination. Third step is the artificial pollination, where that bagged emasculated flower, after the stigma get receptive, the pollen grains are extracted or collected from the male parent flower and dusted onto the stigma of this flower and carried out the pollination, then rebagged over them. Then it is rebagged to allow them to fruit formation. So here third step in that artificial pollination, where that emasculated flower which is bagged over the butter paper over them, that is pollinated after the stigma get receptive. After the stigma get receptive, the pollen grains are collected from the male flower parent and dusted over the stigma, and again it is rebagged to allow them fruit formation, to allow the post pollination events as well as the fruit formation. Till the fruit formation it is rebagged. So this is about the 3 steps involved in artificial hybridization. So this artificial hybridization technique used in the unisexual flowers, where there is no requirement of emasculation process, because you know that staminate flower is different in male parent and the pistillate flower is formed in the another parent. So there is a unisexual plants. In unisexual flowers there is no need of emasculation. They directly bagged the flower to avoid the floral bud, to avoid the contamination of stigma. So emasculation flower, anther is removed from the bisexual flower bud of female parent. After that we can bag that emasculated flower of female parent covered by a bag which is made up by usually butter paper to avoid the contamination of stigma from unwanted pollen grains. After that when the stigma get receptive, the collect the pollen grains from the male parent flower and dust it over the stigma of this female flower, and again it is rebagged and allow them to fruit formation. This is in the artificial hybridization technique. This technique is also used in the unisexual plants where the emasculation process is not needed, where the flower is floral bud is bagged, the female floral bud is bagged by butter paper, and the artificial pollination is carried out after the stigma get receptive, and they again rebagged and formation of the fruit formation is allowed. So this is about the artificial hybridization technique. Then we discuss the another bit, that is double fertilization. Double fertilization is a technique it is a technique where after the entering of the pollen tube into the synergids, what happens? What is the fertilization will take place? Why it is double fertilization? We will discuss here. Double fertilization. So double fertilization after the entering of pollen tube, of one of the synergids of the embryo sac, this pollen tube will releases the male gametes, two male gametes in the cytoplasm of synergid at micropylar part. So the two male gametes which is the entering into the synergid cytoplasm, one of the male gamete will move towards the egg cell and fuses the egg nucleus, and that is called as the syngamy. That is called as the syngamy or fertilization, and form a diploid zygote. So male gamete that is one male gamete moves towards egg cell where egg nucleus is fused called syngamy, or it is also called as fertilization. It is also called as fertilization. To form diploid zygote. To form the diploid zygote. So one thing. So another male gamete which is present in this SYNERGID cytoplasm will move towards the polar nuclei which are present in the central cell and fuses this polar nuclei to form the triploid primary endosperm nucleus, and that process is called as the fusion. That is, two polar nuclei are fused with one of the male gamete, that is form the triple fusion. So second thing is another male gamete is moved towards polar nuclei and fuse them which are present at the central cell. Called triple fusion, and triple fusion present at the central cell called triple fusion, and the formation of primary endosperm nucleus. Shortly it is called as PEN. The primary endosperm nucleus, which is triploid in nature. So another male gamete is moved towards the polar nuclei and fused them, which is these polar nuclei present at the central cell to form the triple fusion. That is called as the triple fusion, to form the primary endosperm nucleus, or shortly it is called as PEN, which is a triploid in nature. So two polar nuclei and the one male gamete fused. There is a formation of three nucleus. That's why it is called as the triploid. Okay, so this is about the one male gamete. When it is fertilized the egg nucleus, there is a formation of diploid zygote, and another male gamete fused with the polar nuclei. There is a formation of triploid primary endosperm nucleus, and that the two times the fusion will take place. One is the syngamy, another is the triple fusion. That's why it is called as the double fertilization. So since two times fertilization, that is syngamy and triple fusion, that's why it is called double fertilization. It's called as the double fertilization. So the double fertilization means one of the male gamete after the entering of pollen tube into the synergid cytoplasm, where they are releasing these male gametes. These one of the male gametes is moves towards the egg cell and they fuses with the egg nucleus. They are fuses with the egg nucleus, and this process is called the syngamy or the fertilization, and they form the diploid zygote. So another male gamete which is present in the synergid cytoplasm will move towards the polar nuclei which are present at the central cell, fuses them. That fusion is called the triple fusion and form a triploid primary endosperm nucleus. It is shortly called as PEN. Since there is two times there is a fertilization takes place, that is syngamy and the triple fusion, that's why it is called as the double fertilization. Now this double fertilization, it is common or it is the event unique for flowering plants. So double fertilization is a unique technique or unique events occurs in the flowering plants. Now the central cell which is present at the central located in the embryo sac, this central cell after the triple fusion becomes the primary endosperm cell, that is PEC, primary endosperm cell, and that is called as the PEC. Again, after this, they develop into an endosperm. They develop into a endosperm, and the zygote develops into a embryo. So zygote develops into embryo, and the primary endosperm cell develops into a endosperm. So what is this endosperm? It is a triploid in nature, means there is a PEN present in the. So this is about the double fertilization. You have to draw the diagram of a fertilized embryo sac. So fertilized embryo sac, after the fertilization, after the double fertilization, what happens in the embryo sac? What is the structure will be changed in the embryo sac, that will be discussed with the help of the diagram. We can draw this diagram like this. So after the fertilization, what the changes takes place in the embryo sac? This is the micropylar end. So this is micropylar end, where we observe the three types of cells, that is two synergids and the one egg cell, which is the larger one because it is developed into a zygote. So this is about this one. Then centrally located a cell will be observed, that is the central cell becomes PEC now, that is primary endosperm cell, that is primary endosperm cell, shortly it is PEC, and this is the PEN, that is primary endosperm nucleus. Then below this here we observe these three are the degenerative, means after the fertilization these cells are degenerate, that are antipodal cells. So degenerative antipodal cells. And what happens to the synergids? Synergids also degenerate. So degenerative synergids, and this is the central cell which becomes a primary endosperm cell which develops into a endosperm after the fertilization, and the PEN, that is primary endosperm nucleus, becomes endosperm, and this becomes a zygote, that is the zygote which will be diploid, it is diploid. So zygote which is formed after fertilization, which is develops into a embryo. So this is about the diagram about fertilized embryo sac. It is a fertilized embryo sac. So earlier class we discussed the diagram structure of embryo sac, that is the matured embryo sac. This is a fertilized embryo sac. After the fertilization, what is the changes takes place, what are the events occurs within the embryo sac, that we will discuss with the help of this diagram. So tomorrow we discuss the post fertilization events, and what are the post fertilization events, that you know that there is a embryogenesis takes place and the zygote development will takes place. So that is we will discuss in next class.